Okay, so now first of all, when we talk about gene mutations, gene mutations highlight they are random and they're spontaneous changes. Okay, random and spontaneous. Basically, we don't know when they happen. Okay, so it can happen at which level? DNA level. Basically, a particular gene. Okay, so we actually can change the number of nucleotide. We can change the sequence of nucleotide base of a gene. So therefore, gene mutation also known as DNA mutation also known as a gene mutation. Are you clear? So it only affects a particular gene only. Okay. Or the mutation may take place to change the number or the structure of chromosome. Number, for example, Down syndrome. We know about Down syndrome. If you look at Down syndrome individual, you will check the DNA. Sorry, you check the number of chromosome. They have 47 chromosome rather than 46. Because the chromosome 21, they have additional of one chromosome 21, or we call trisomy 21, trimin 3. Okay, so it means that at the chromosome number 21, they have 3 chromosome 21s rather than compared to normal persons, we have 2 chromosome 21. Okay. Now, how this gene mutation can take place? We have 4 ways for gene mutation to take place. The first one is the most common one, and it takes place every moment in our body, which is spontaneous mutation. And this spontaneous mutation always takes place during DNA replication. So it's slightly actually different from what we have learned previously. Because when we learned previously, we said that DNA replicates semi-conservatively, correct? Okay, so it means that the parental strand will act as a template. So when the parental strand acts as a template, it should be correct because, for example, if the parental strand is A, T, T, C, C, G, for example, DNA polymerase should add correct ones with T, A, A, G, G, C, correct? So this is ideally it should be, or it must be copied, okay, exactly. Are you clear? Correct or not? Okay, correctly in this case, but our DNA polymerase sometimes make mistake. So for example, okay, instead of G, it may accidentally, okay, for example, add a T here. So it means that mutation take place. So this kind of mutation is spontaneous mutation, always take place in our body, but we won't feel it because our body do have a mechanism called DNA repair mechanism. So during this DNA repair mechanism, because the rate of the spontaneous mutation is quite low, basically our body do have sufficient time, our cells do have sufficient time to do the correction by doing this what we call the DNA repair mechanism. Wrong one, we add a new, we rectify it. Are you clear? Okay. Now, second, the mutation due to the error-prone replication bypass, okay, sorry, or naturally occurring DNA damage. So sometimes our DNA do damage here and there. Okay, so during DNA replication, for example, when we do have this our DNA, so some part of DNA here actually damaged. So when DNA polymerase add, it may bypass this region and then continue. So it means that you can see that small segments of the DNA is not copied or it's not, replicated. Are you clear? Okay, it becomes shorter, the DNA. So therefore, this one also can cause the mutation, a short segments of DNA actually remove. Are you clear? Okay. Or error introduced during the DNA repair. Now, this is funny part here is DNA repair by right. The DNA repair mechanism should correct it, okay, make it correct, right? For example, just now we say that C here, we should repair and become the G, correct or not? But instead of making it G, now from T, it becomes, okay, for example, it makes it C. Are you clear? Basically, it do the double error. So this can happen. Or the last one, it calls induced mutation by Mutagen. Now, what's induced mutation? Induced mutation basically means that you must have certain chemical, okay, or a particular types of the agents that cause the mutation take place at high rate. Now, compare number 1 and number 4. Spontaneous mutation take place always at a very low rate. Are you clear? So our body repair mechanisms do have sufficient times to actually rectify it. But when you talk about this, induced mutations caused by the mutagen, Mutagens means that agents that cause the mutation to take place. So it will take place at very high rate. It's so high until our body repair mechanism may not have sufficient time to rectify it. We don't have the time to check it, and it passed down to the descendants of the cells. So therefore, mutation actually is there. Are you clear? Okay. So mutation arises spontaneously at a low frequency due to the chemical instability of the purine and pyrimidine and the error during this DNA replication. So it means that we add the wrong one, okay? So induced mutation are alterations on the gene after it have come to contact mutagens and the environmental causes. It can be either physical or chemical. So the idea here is to increase the frequency of the mutation or increase the rate of mutation. So table 10.13 show you guys type of the mutagen, but this is for your information only. You can read about it. If you're interested to know more about the mutagen or chemical mutagen, feel free to read about this. Okay, we have few classes of the chemical mutagen. Okay, we have ROS, reactive oxygen species, where it always attack the DNA, damage the DNA. We have deaminating agent, alkylating agents, and the intercalating agent. Those are the agents or the chemical, they have the ability to damage our DNA. So when they damage the DNA, it will cause the mutation to take place, okay? So the first class of the mutagen is termed as chemical mutagen. The second class of the mutagens is known as the physical mutagen. So this one, I think you guys are familiar with this. When we talk about X-ray, gamma ray, okay? So those are the radioactive ionizing radiations, UV. So those are the physical mutagens. So they have the ability against to damage our DNA and cause the mutation to take place, okay? Now, when talk about UV, people always think about, we talk about the ionizing radiations. So people always think about this. Can they cause cancer or not? The answer is yes. Okay, but be careful of these two terms, guys. Carcinogen versus mutagen. Are they the same? Are they the same? Carcinogens and mutagen. Carcinogen basically means that's the agents or chemical or any agents that cause cancer. Mutagen is the agent that causes mutation. So we do have some chemicals belongs to both groups. They cause the mutation of the cells and result in the cells, right, having high rate of the cell cycle. So basically means from 1 become 2, 2 become 4, it multiplies so fast. Yes, it may be. Okay, for example, ethidium bromides that we're going to learn about this ethidium bromide. When we talk about ethidium bromides here, ethidium bromide belongs to both classes. It can be a mutagen because it already mutates the DNA. It can cause cancer as well. So that's why it also classified as the carcinogen. Okay, but some agents, they only cause cancer. They don't cause mutation. It is there, okay, to make your DNA or to make your cells to multiply faster, but they don't have any effect on your DNA. So this kind of agent is termed as carcinogen. Are you clear? Okay, but some mutagen, they can cause cancer, but not all mutagen cause cancer. Some mutagen actually cause the other type of disease. For example, sick anemia is not cancer. Hemophilia is not cancer. PKU is not cancer. Are you clear? So don't put an equal mark there between these two words, carcinogens versus this mutagen. Don't put an equal mark there. Are you clear? Okay. Now, in multicellular organism with the dedicated reproductive cells, okay, so we can subdivide it into the germline mutations or somatic mutation. So it depends on where is this mutation actually take place. If the mutation take place at the testis or at the embryo or at this ovary of the woman, then what will happen here is, because the ovary contains the egg cells and the testis contain the sperm cells, so any mutation will be passed on to the next generation. So inherited. So this kind of mutation is quite dangerous. So particularly when female, when you go for the X-ray, CT scan, those what we call this ionizing radiation type of the scanning. So the doctor will ask, are you pregnant or not? Because this will cause the problems, okay? I mean the mutations to the embryo if let's say any pregnancy take place, okay? So there's more to the germline mutation. So a somatic mutation means that when we talk about somatic mutation is mutation that takes place on the somatic cells. For example, skin, mutation on the skin, mutation onto your liver, mutations onto your heart cells. Those kind of cells are not reproductive cells. So it means that it won't pass on to the next generation. It only stop at your generations only. Are you clear? Okay, so that's kind of mutations called somatic mutation. Okay, so let us look at the mode of mutations. We have three modes of mutations here. So be careful when talking about mode of mutation versus the effect of the mutation. When talking about mode of mutation means that how the mutation actually takes place. Okay, so we have three kinds. We have what we call the base substitution. Even though we use the word base, but you have to understand that we don't change the base only, we change the entire nucleotide. Are you clear? So by right, it's ATTCCG, now become ATTCCA, for example. So this kind of mutation is termed as the base substitution. We exchange one nucleotide with another nucleotide. By right should be A, we change it to T, or C, we change to G, for example. So this kind of the mutation, the mode of mutation is known as base substitution. We substitute the base. Are you clear? Even though we use the word base substitution, you have to understand that in actual case, we don't substitute the base only, we substitute the entire nucleotide. Okay, so there's base substitution. Then we have another two quite similar. One is to add, another one is to remove. Okay, so we have the base additions, means we add additional nucleotide. Okay, or base deletion, where we remove the sum of the nucleotides. It can be one, it can be more than one. Are you clear? Okay, so now based on this, please complete table 10.14 by indicating, say, the modes of mutation now. Okay, so look at the first row. We talk about the process involved the exchange of a single nucleotide for another. Not necessarily may be single, it may be more than one. Okay, so in this case, exchange. So it must be the base substitutions. Okay, so base substitution. So the most common one is called transitions. Transitions, basically, we exchange purine for purine. Now, what's this mean? So let's say, for example, so we have, okay, T, A, for example. Okay, so when you add, by right, it should be T, now you add the A, by right, it should be A, for example, ATT, kind of the complementary strands. Okay, the DNA polymerase. But instead of adding A, now it adds G. Can you see that? It adds G. So initially A is purine. G also purine. So this kind, when they change A to G, G to A, in this case, or C to T, T to C, this kind of exchange is called transition. Are you clear? It's called transitions. So transition is quite common. Are you clear? Transition is quite common compared to the transversion. Okay, transversion. Transversion, basically you exchange purine with pyrimidine. So what will happen here? Now try to imagine. Purine, longer, right? 2 rings. Pyrimidine, 1 ring. Are you clear? So means that you try to imagine if originally is the pyrimidine, now you exchange it with purine. So means that you can see the DNA now bulging. Cannot. Try to imagine purine's longer, 1.2. Pyrimidine is 0.8. So maintains the DNA diameter at 2.0 nanometer. Cannot. Okay, but now instead of pyrimidine, we change it to purine. So when you have 2 purine together, you have the bulging area there. So these special parts will be easily detected by DNA repair mechanism. So DNA repair mechanisms can easily identify and then do the repair. So this kind of mutation actually is very, very rare. It's not so common compared to the transition. Or when you have the pyrimidine, okay, by right you should add the pyrimidine. Sorry, by right you should add purine, you add pyrimidine. So when 2 pyrimidine together, you can see that it form a groove like this. Okay, the groove. So this groove basically will be against allow the DNA repair mechanism to identify it and do the rectification. Okay, so that's why they are less common. Okay, now what's the effect of mutation? Effect of mutation, it can be the silent mutation. Now what's mean silent mutation? I thought silent mutation means that this mutation, okay, happened, but no effect. Thanks to what? Thanks to our triple code. You check back the triple code, you can realize that some amino acid, they are coded by more than one code. For example, AAA, for example, and AAT, both of them code for the same amino acid. So even though DNA mutation take place, it doesn't change the type of amino acid. So mutation, you will check DNA, yeah, mutation take place. But check protein, no wrong. So this kind of mutation is termed as silent mutation, which codes for the same amino acid sequence. Okay? Then we have these two called missense and nonsense. Okay? So missense basically means that we exchange the amino acid. For example, when the mutation take place by right at the position, for example, sickle cell anemia, beta polypeptide. So still remember at the 6th position, originally is a glutamic acid, which is polar, which is a hydrophilic, but changed to valine, correct or not? So this kind of mutation, the effect of the mutation is known as the missense mutation. So the original length of the polypeptide still remain the same, but one of the amino acid now we exchange with another. So it depends on the severity. So some protein may still function, some may have the reductions in terms of their effectiveness or the functionality, or some of them may not even have the function at all. Are you clear? So this is called missense mutation. Basically, you still get back the whole line. Okay? Now, sometimes this base substitution may introduce what we call the stop triplet or stop codon. So means that by rights, my amino acid, polypeptide may have hundreds amino acid long. But at, let's say for 20, by right, so go 21, because a mutation introduced the DNA triplets which code for the stop. This means our translation process will stop prematurely. So the polypeptide becomes shorter or truncated. Okay, we use the word truncate or truncated. Truncated basically means that shorter. Later I will show you guys, okay? Truncate means shorter, okay? Now I want to highlight these two, guys, silent mutation and missense mutation. These two is very special for base substitution. Only base substitution can cause silent mutation and missense mutation. Base addition and base deletions cannot. Are you clear? Okay. Nonsense mutation, right? For nonsense mutation, it can be base substitution, it can be base addition, it can be base deletions. Are you clear? Okay, Now next. Second row, one or more extra nucleotides are added into the DNA. One or more extra nucleotides are added into DNA. So this one's very, very simple. It's a base insertions or base addition. Okay. So it results in what? It results in the frame shift mutation. Now what's the frame shift? Reading frame change. Now, what's mean reading frame? Okay, so now look at this. When we have the sequence AAA, TTT, CCC, GGG. If we add another A here, the reading frame now become what? Oh, sorry. If we add another A here, so the reading frame become AAA, ATT, TCC, CGG. So means that the entire reading frame change. Can you see that? Are you clear? So it could, or the whole thing, most likely, when after the mutation parts, the whole length, after that, all amino acid might be wrong. Are you clear? So this kind of mutation is called frame shift mutation. Okay, frame shift mutation. So frame shift mutation actually is special for base additions and base deletion. Because base substitution won't cause frame shift. Frame shift only can be caused by base insertions or base deletion. Okay, you look at this. When you remove, so it causes the same types of the results. Okay. Another one called nonsense mutation, because when you add or you remove, when you add, okay, the nucleotide or remove a nucleotide, you accidentally, the process of mutation accidentally introduce the DNA triplet or stop triplet. So means that against the polypeptide actually stop. I mean, the translation process stop halfway. Okay. So therefore, you get a truncated polypeptide, means that the your polypeptide become shorter. Okay. So now we look at this single base substitutions. Yeah. So in this single base substitution, this is a wild type, guys. There's something special about biology. When you talk about wild type, not means that you're wild. In this case, wild type means normal. Are you clear? Okay. In biology, we say about in bio, when you talk about wild type means normal. So in terms of this normal, you can see that the TAC codes for the methionines, TTC is lysine, AAA code for phenolphthaleins, and CCG code for glycine, and then ATT stop. So this polypeptide, actually it means just a makeup ones. So therefore have 4 amino acids with a sequence of methionines, lysine, phenolphthaleins, and glycine. Okay, MLPG in this case. Okay. Now, what will happen if now, okay, TAC, TTC, AA, CCA instead of CCG. Now, you check back the DNA triplets, okay, codes there. You'll realize that CCA, CCG, both of them code for glycine. Okay, both of them code for glycine. So in this case, we do know that it's a silent mutation. Why silent mutation? Because the base substitution here didn't cause the change in the sequence of the amino acid in this particular polypeptide. Clear or not? Okay, so methionine, lysine, phenolphthalein, and glycine. You check, you still get back methionine, lysine, phenolphthalein, and also glycine. So state the effect of the mutation. So how you write this? So for me, I will write both. Okay, so it's a base substitution. Results in silent mutation. Okay, so base substitution result in silent mutation. Okay, now next, second case. So you can see that AAA, TCG. So in fact, CCG now change to TCG, base substitutions. Are you clear? So one single nucleotide substituted here. Instead of, okay, C, we have T. Now, if you compare, if you check back the DNA triplets in the notes, 10.3, triple code table, then you realize that TCG code for serine. So if you compare, original is glycine, now serine, but the length still remain the same, correct or not? Are you clear? When the length still remain the same, so it means that we have exchange of one amino acid. This one is termed as the missense mutation. See, missense mutations. So we say that base substitution results in missense mutation. So as I say that, okay, silent mutation and missense mutation specific for, is a result specific for the base substitution. You won't get this missense mutation and also the silent mutation from the additions or deletions. Okay. Now next. So in this case, so instead of TTC now become a ATC. ATC is a stop triplet. Can you see that prematurely stop? Can you see that? Prematurely stop. So your amino acid from 4 become 1 only. So this is called nonsense mutation. So what is the effect? So we say that's a base substitution results in nonsense mutation. Base substitution results in nonsense mutation. Okay, so now we look at the base addition and deletions. The effects almost the same for base additions or base deletions. Okay, so again, the same wild types, not a mutant. The wild types, you still have the same sequence of the amino acid. We have the methionine, lysine, phenolalanes, and also the glycines, and then we have the stops here. So 4 amino acids to make up these polypeptides. Okay. So now you look at this. If we have extra A, and so happen that when put the extra A here, by right, it's TTC now become ATT. ATT is a stop triplet. So again, you can see that's our DNA. Can you see that? It's the DNA now. Okay, stop DNA. The polypeptide synthesis, or they call it the translation, stop prematurely. So this kind again is a nonsense mutation. Okay? So we see that this is additions. So base additions. Okay, results in a nonsense mutation. Okay. Or when you remove, okay. So by right, the reading frame is AA now become AAC. Because A removed already, so the rest will be different. Cannot. We have methionine, lysine, okay, phenolalanes, and also the glycine. But now can you see that the frame shift actually take place? The rest of the amino acid sequence will be wrong. Cannot. Okay. So this kind of mutations, okay, is base deletions. In this case, it can be base additions or base deletions. Okay, results in frame shift mutation. Okay. Okay, now next. Before we proceed to the next example, okay, so I'm going to show you guys these 2 examples here, or 2 scenarios. So we have the base deletion. So we have 22 base deleted, or nucleotides base deleted. Okay, 22. Oh, sorry, not 22, my mistake. 21. Okay, we have 21 nucleotide base deleted scenario, case number 1. And case number 2, we have 14 nucleotide bases or base pair deleted. Okay, so case 1, case 2, both of them very severe, but which one more severe? (21) deleted more severe or 14 more severe? Yes. Any suggestions here or any idea? 14 more severe, right? Why? 14 because it's not a multiple of 3. When it's not multiple of 3, so it means that you can cause frame shift. If you look at this, if 14 nucleotide bases deleted, confirm it cause frame shift because not multiple of 3. But how about 21 nucleotides? 21 is multiple of 3. So basically, after the deletions of these 7 amino acids, the rest will be the same. But both cases, they are severe. It's just that which are more severe in this case. So on paper, case number 1 will be less severe compared to case number 2. Are you clear? So if we go to this, you can see that this we call the trinucleotide deletion, guys. Trinucleotide deletion. And you can see that you miss 1 amino acid, but the rest still remain the same. Phenolalanes and glycine, so missing the lysine here. Correct? Okay, so we say that this one is a trinucleotide. Deletions, oh, sorry. Results in the missings of one amino acid, but the rest they're the same. This is not frame shift, guys. No frame shift at all in this case. Are you clear? Frame shifts only occur when you have one or two, okay, nucleotides deleted, okay? But in this case, no, because we try nucleotides at the end, we can see that we still get back the same amino acid sequence. It's just that one amino acid missing. Are you clear? So severity is not so high, but again, it doesn't mean that you won't get any impact. The impact is still there, but it won't be so severe compared to the one or two nucleotides deletions or additions. Okay. So we use one example to study about this gene. What call this gene mutations by using the sickle cell anemia. So we do know that sickle cell anemia. First of all, we talk about this hemoglobins, right? When we talk about hemoglobins, we do know that they have 4 polypeptides, correct? Okay, 4 polypeptides, 2 alpha and 2 beta. But at the beta, polypeptide change there. So for normal or wild type, say we say wild types at the 6th position, highlight guys, it's a glutamic acid. So this glutamic acid is acid, right? The glutamic acid, so it's a hydrophilic. So it maintains the solubility of the hemoglobin. Okay, but because of the mutation take place, a single base substitution from CTC replaced with CAC. Okay, CTC replaced, okay, the T replaced by the A, the thymines. Okay, so this amino acid sequence now, instead of having glutamic acid at the 6th position, now we do have the valine at the 6th positions, and the valine is a non-polar. Okay, the R group is non-polar, right? Hydrophobic, so decrease the solubility. Therefore, the cells tend to become sicker due to the polymerizations, or we call it these aggregations of, also the fibrous aggregations of the hemoglobin. So in this diagram, you can see that, guys, from the T become A. So therefore, glutamic acid now change to valine. It causes the sickle shape of the cell. Okay. Now, generally, when this question asks about the mutation, what is the effect of the mutation in general? So we have to answer in 4 levels. The first level we talk about at the DNA level. So you're going to change the sequence of nucleotide. Are you clear? You're going to change, right, the mutation. Number 2, at polypeptide. So it will change the primary structure of the polypeptide. Means that the sequence and the type of amino acid will be different, particularly when we talk about the frame shift. Okay? And number 3, we talk about the folding of polypeptide. So it changed the shape of folding, the way of folding. So it means that it affects tertiary or even quaternary structure. Okay? It may change the tertiary or even quaternary structure of the proteins. And number 4, we talk about the functions. So it may reduce the functions in general, or it may have no functions at all when talk about the mutated. Okay, when the mutation process take place, you may see that the proteins that form after folding everything may not have function at all. Okay, we look at these sickle cell, the sickle cell hemoglobin still have function. They still can carry a group, right? But it will cause the problem called the disease. Okay, but some protein may not have functions at all. Right? Particularly we talk about the nonsense mutation, the protein become shorter. Okay, so please do remember those mechanism. Okay, because we're going to look at this again when we do go for the A2. Okay, where we're going to look at some diseases that due to the mutations. Okay, so with this, I've done for this topic of the recording.